Hi, I'm Brad Power, and this is the Cancer Patient Lab, and we're honored to have today with us Padman Vamadevan, who's in London, and Travis Christofferson, who's in Lake Placid, New York, although he's normally in Colorado. And they're going to be leading a discussion with us about their approach to precision oncology and some of their services. Just a few housekeeping things before we get started. There are three. One is this is for information purposes only. This is not medical advice. We try to arm our patients and caregivers with information they can take to their medical team. Second, this is all going to be made public. So if you're concerned about having your image shared or your name or anything else, you can change your name, turn off the camera, and not say anything. And then finally, we are a nonprofit 501c3, and we depend on the kindness of people who make donations to bring you services like this webinar. And you can do that very easily on our website where there's a prominent donate button. And with that, I'll turn it over to Padman and Travis.
Thank you very much, Brad. I'm just going to share my screen if that's okay. Hopefully that's working for everyone. Perfect. Thank you, Brad. So yeah, thank you so much for inviting us here today, Brad, and to all of you for showing up. I know you're all busy people. So my name's Dr. Padman Vamadevan. I'm the co-founder and chief medical officer here at Astron Health, based in London, although we have a very cross-Atlantic team. I'm here together with my co-founder, Travis Christofferson, and also one of our founders associates, Raphael Leong, who's also on the call. And today we just wanted to introduce you to a new paradigm that we believe could fundamentally enhance the way that clinicians can use molecular profiling to develop robust, personalized treatment strategies for patients like yourselves. So having watched a few of the CancerPatient lab videos, which I must say I think are a phenomenal source of education for patients and clinicians and scientists alike, actually. We know that patients like you understand the complexity and heterogeneity of cancer. You know also that conventional oncological treatment options can sometimes fall short. And the reason I think that we built Astron was to try and give each patient and each doctor the confidence that they are doing absolutely everything possible to personalize cancer treatment based on molecular science. So just a bit of context about Travis and I. So before Astron, we spent over a decade together at a biotech that worked trying to develop real-world clinical studies looking at the potential of drug repurposing as adjunctive treatments in cancer, so very much in addition to standard of care. Sometimes we saw the power of these approaches, but we also saw their limits when they were used generically. Some patients did better than expected, but unfortunately, too many plateaued, too many relapsed, and too often we felt that we were treating the disease using overgeneralized principles rather than targeting the specific vulnerabilities of that individual patient's cancer. And an example of this was a study that we published in 2019 in glioblastoma multiforme, where the top line was that in 96 patients, the median survival of those patients was almost double expected. But unfortunately, when we looked a little bit deeper at the patient cohort, that was driven by about 40% of extreme responders. And we really weren't satisfied for that. We wanted to do more for the other 60%. And we thought that actually the thing that we were missing was the personalization of our approach. We were using the same standardized adjunctive protocol for every single patient, and some were benefiting and others weren't. And trying to translate that overhang where we were trying to sort of match all the research data that was out there into personalized treatment protocols was something that we just couldn't keep up with. There are thousands of studies being published every year, hundreds of molecular vulnerabilities in each tumor type. We were barely scratching the surface, to be quite honest with you, and that's why we built Astron. It's in its core a bioinformatics platform powered by large language models that becomes your personal cancer research team. It's designed to be built for patients and their clinicians. Clinicians need data, they need transparency, they need clarity, and they need all of that in real time whilst you're in the consulting room with them. And that's what we try to offer. So in terms of the core of the problem, I think it's this, that we have, as a society, really invested really heavily in molecular testing in cancer over the last decade. And a lot of the talks that you've done at CPL have been around groups doing phenomenal work to improve outcomes and really sort of amazing solutions. I saw the call you did with Laura Tower and the team at Vivan last week. But unfortunately, with the core molecular testing that often appears in the consulting room, most of that testing in terms of the report ends at a diagnostic stage. The report gets printed, it tells the doctor and the patient what mutations might be present, what genes might be overexpressed. It might list a few FDA-approved drug options or clinical trials that may or may not be available in your geography, and that's it. It's really not uncommon for a next generation sequencing report to be brushed over in a clinical consult because there just isn't the clarity or the optionality or the time to do the research to enable that clinician to actually do anything about it. There's a huge technological overhang, I think, that clinical medicine struggles to keep up with. And that's really where we begin. We try to take the tidal wave of raw molecular data, DNA, RNA, proteomics, microbiomics, immunomics, everything, and try and translate that into a clear curated treatment strategy based on the breadth of available peer-reviewed data. And the platform really is built to do one thing, to turn all of those nuggets of raw molecular data into personalized, low toxicity intervention plans that clinicians can use today for their patients. The other part of the problem in precision oncology, in my opinion, is that it historically has been viewed as expensive, inaccessible, and often inefficient. And I appreciate that lots of you are patients who are trying to solve the riddle of their own disease at an N=1 level in real time. But I think it's also important to take a step back, and it's pertinent to remember that many cancer patients globally have no evidence to testing or precision oncology interventions at all. They never even get to the starting line. And so for us, I think our priority is to try and change that baseline for everyone and to do so at scale. And to do that, we've really tried to hone and focus our approach on what we can do today using interventions that are already available and being prescribed safely. We're just trying to do it smarter and more precisely. So I'm going to try and do this very quickly for time reasons, but we've made huge strides in understanding cancer genomics and molecular variations. There are more than 750 identified cancer-driving mutations as defined by COSMIC, but unfortunately only around 50 to 53 are currently druggable with FDA-approved cancer therapies. And that's the core problem with precision oncology today. Our ability to test far outpaces our ability to act. Creating a new precision drug takes somewhere between 10 and 15 years and costs somewhere between $1 and $4 billion. And drug companies want to de-risk their R&D, so often concentrate on creating drugs that hit molecular targets that are high ticket. And yet there is a vast body of literature pointing us to ways to modulate many of the currently undruggable mutations using already available therapeutics. And Travis is going to talk about that a little bit later. The data is there. It might not be there in the format, but people need to see it. It's scattered across hundreds of thousands of publications. And there is, again, a massive paucity of empirical outcome data. The other part of this that I wanted to address is that there is a lot of information out there in groups, curated by groups without full understanding of the scientific process. And that attracts patients often to take anything and everything that has any putative anti-cancer activity. And this can be really risky. I see this every day. Patients taking 30, 40 different things. It's not the way to build evidence or credibility around these approaches. So we've built Astron to absorb the complexity, synthesize the research, return something that clinicians can use in real time in the consulting room. And our approach is already moving the needle, I think. We feel like our bioinformatic approach can hit around 70% of COSMIC-defined cancer genes to some extent, and that will only improve as the database scales and more research is published. So I'm going to move over this one pretty quickly, but it's really important to know that we are really trying to build this in a practical way so that we can plug into every clinician's workflow seamlessly. We want to be seen as a research partner. We're not a testing company. We are not the end prescribers. We're not trying to take anyone's place here. We're just trying to do the research that some of you are doing, some of your clinicians could do if they had a spare 30 hours per patient to do it. And we're trying to do it in a really data-driven, focused, robust way for each patient. So once we receive a patient's NGS report, we can run a deep analysis using our AI-powered engine. It's not a black box. It's a transparent, literature-supported, mechanistically scored recommendation engine. And the outputs are then manually curated by our scientific team thereafter. So it's not just a black box. And then within 5 to 7 days, we deliver a research report to your medical team that prioritizes interventions based on the strength of the scientific evidence, the mechanistic plausibility, the disease context, the tissue, the stage, the mutational burden, the drug safety profile, tolerability, known interactions. So we don't just give people a list of molecules, we give doctors a treatment hypothesis ranked by strength, supported by references, contextualized to the individual's case. And the key to this as well is that we're really extending the breadth of interventions that we're exploring. So we're looking across all drugs, oncology drugs, non-oncology drugs, nutraceuticals, even lifestyle modifications, anything that has data in a cancer setting. And we are trying to really democratize access to precision oncology by being broad about this. So our methodology doesn't depend on inventing new drugs, but rather matching existing therapeutics intelligently to each patient's specific molecular vulnerabilities. So it's not, to summarize, I suppose, it's not about boutique science in advanced, hard to treat individual cancers at huge individual cost. What we're really trying to do is to deliver public health impact at a global scale using technologies and therapeutics that are available now. I'm going to brush over this one. I'm also going to brush over this one because I think Travis is going to speak to it, and I'm just a bit wary of time. So we want to just go over a quick case report, Trav, if that's okay.
Yeah. How much time should we shoot for here, Brad? About 5, 6, 7 more minutes?
That's all fine. No worries about the time.
Okay, good. This will be quick. So what this is, everybody, this is a sample case report from a redacted patient with colon cancer. And this will show you what the report looks like. And just to give you an example of our methodology. So the genomic reports with this patient came from Tempest and I believe Datar Genetics. And so you can see on the left-hand side, those are the gene mutations within this patient's cancer. We then identify, using a hierarchy of data, the most valuable target presented with each of these mutations from bioinformatic sources. We then expand that to drug repurposing and find compounds that will inhibit that target. And we use a number of strategies. You can see in the target type, we can go with downregulation of upregulated genes. We can go with direct inhibition of activated oncogenes, or we can use this concept known as synthetic lethality, which is a complex name for a simple concept, strategy in therapeutics, where when a cancer cell has a mutation, particularly in tumor suppressors, it will rely on a compensatory pathway. And so if you target that compensatory pathway, it results in a non-viable state for the cancer cell, where the healthy cell is fine because it doesn't have the mutations. So I'm just going to talk through just a couple of these because there's a number. You can see this patient has an NRAS mutation, which forms a synthetically lethal relationship with MEK1 and 2. Next slide, Pat. So when you go to the bioinformatic drug databases, you can see that this natural product, myricetin, is a strong inhibitor of MEK1 and 2. And it's been shown in lots of in vitro and animal studies to have anti-cancer activity. In this particular case, we're looking for direct, specific targeting of MEK, which it does as good as the chemical compounds that you buy to be inhibitors of MEK1 and 2. Okay, next slide, Pat. This one's interesting. We see a relationship with PIK3CA, a very commonly mutated gene in cancer in general. I think about 15% in colorectal. You have two options with metformin, a downstream inhibitor, and believe it or not, caffeine is a good direct inhibitor of PIK3CA. This caught our attention. It probably like it does yours. It's very curious. And you go back through the literature and you can see that caffeine in the drug databases is highly pleiotropic. And it's been known since the 70s. It inhibits ATM and ATR, DNA repair genes, and has been known as a radiosensitizer for a long time. So we got curious about, there's a strong relationship between PIK3CA inhibition and ER estrogen receptor inhibition in breast cancer. This presents a really good synthetic lethal relationship. So we wondered if there was any clinical data on that. And this really speaks to the predictive power of this approach. This was, I think, around 2015, a headline that coffee consumption could reduce the risk of reoccurrence in a certain subset of breast cancer patients by half. And okay, next slide, Pat. And so that came from a study in Sweden on over 1,000 women. And they were simply asking the question, what is the impact of coffee consumption? They didn't have any mechanistic insight, because people are always curious about coffee consumption because it's so ubiquitous. And what they found, the study initially found no response, but when they broke out this subgroup of the ER positive women on tamoxifen, so they're inhibiting the estrogen receptor and PIK3CA with caffeine, you see this dramatic reduction in recurrence and death. So it's just a validation, a clinical sort of study validation that we would suggest is due to this mechanistic relationship. Okay, next slide, Pat. Okay, I'm sorry, what are they? I'm getting a message blocking my screen. Okay,
APC.
let's skip the APC and go to the very last one, because I think everyone understands the methodology. The very last one is an interesting one. Now, this is a relationship between PIK3CA and a gene called PTGS2, which is the gene that encodes COX-2, the very, very famous inflammatory enzyme that all the NSAID drugs target, like ibuprofen, aspirin, and so forth. And celecoxib it falls in that class of NSAIDs. So this is sort of the higher tier evidence that we are sort of shocked that clinicians don't have access to. Okay, next slide, Pat. So with a particular mutation, the PIK3CA, there was a study done, a recent study, a randomized placebo-controlled phase 3 trial. This was a huge study done at 654 community centers and hospitals, over 2,000 patients. The study basically was people with stage 3 colon cancer that had surgical resection followed by chemotherapy. Were split into two groups. One received just that standard of care. The other received exactly that standard of care with celecoxib. And when they broke out the patients with PIK3CA mutations, they demonstrated a 50% reduction in death. Now that's incredible. That's, massive. And when you look at this through the lens that we look through with the risk reward, if that drug, adjunctive drug, was adding a significant amount of toxicity, then you question whether the data has the veracity enough to go forward. But you can see there was no significant toxicities introduced from it. There was no increase in adverse events from adding it. So it's a very safe thing. And with through that lens of risk reward, we think patients and doctors should have access to this type of data. Okay, last slide, Pat. And this is just the last slide that I'll end with. And this is just to emphasize the problem that we are trying to solve. This is an article in the New York Times from 2008, oddly enough, written by Billy Beane, Newt Gingrich, and John Kerry. Billy Beane is a manager for the Oakland A's. I don't know how many of you have seen the movie Moneyball, but what he did there was take a complete data approach to player selection. They had, I think, the second smallest budget in the league at the time, 3 times less than the New York Yankees, but were able to take the team to win the championship, not the World Series, but their league championship. The way they did that was just simply sifting through data to see what was undervalued, what people were missing, and that's exactly what we're trying to do. The quote from this article that I'll leave you with: remarkably, a doctor today can get more data on the starting third baseman of his fantasy baseball team than the effectiveness of life and death medical procedures. Studies have shown that the most healthcare is not based on clinical studies of what works and what does not. It's not be it a test, treatment, drug, or technology. Instead, most care is based on informed opinion, personal observation, or tradition. And most people give Hippocrates, know the Hippocratic Oath, first do no harm. But the first quote from Hippocrates was, a doctor's judgment matters more than any external measurement. And that sort of ethos has carried through medicine, where doctors are viewed as sovereign entities upon their own. We've reached a time today where there's just too much complexity and too much being missed in the realm of data. And that's the void that we hope to fill. Okay, Brad, that's my end. Sorry for being so verbose.
Are you done too, Padman, or did you have more?
No, I'm done. I think I spoke for long enough. There was a couple of slides that I skipped over, but I'm sure we'll arrive at that during the conversation.
Right. Okay, so for those of you who've been here before, the way that we solicit comments or questions is through the chat. Oh, and there are a couple in there now, as well as through the raise hand feature. And I will help moderate that. So Padman and Travis, you don't have to be following all that. I'll read the first one here from Russ. Have you seen this? And then it's a link to something from cell.com. It looked like a nice genetic analysis using Stampede as a basis. Stampede being I guess, a clinical trial.
Yeah, and it's really interesting actually. Sorry, I can't act for some reason. I'm going to stop my share because I can't actually see the chat. I can now. Stampede's an interesting one actually because there was an arm of Stampede where they used metformin as an adjunct, and they used it. It's interesting when you look at the slides from the study, and they used metformin very much as, you know, they recognized that enzalutamide was going to cause all manner of metabolic dysfunction and raise glucose and insulin and all sorts of things. But actually, it was interesting in the slides that they talked about metformin in an anti-cancer drug as well as a metabolic optimizer for those patients, which was interesting. And I think I've got to be honest, I'm not completely up to date with Stampede, but when I last checked and looked at the subgroup analysis of that particular strata of the Stampede, they didn't show a particular effect of just metformin. I think the really interesting thing here is when we look back now at some of the studies that are being done, even clinical trials. The study that Travis spoke about with celecoxib was actually a Natera-sponsored trial, which was a subgroup analysis of, they were using Signatera as an early detection tool. And that was actually, there was a study in colorectal cancer looking at all comers, so not stratifying by PIK3CA mutation. And it basically found that adjuvant celecoxib had no impact on overall outcomes at all. And I've had lots of patients that have come to me taking celecoxib, and their oncologist has said, there's no benefit for this at all. Here's the clinical trial. And what we're now doing is saying, well, actually, in this subgroup, it will benefit. So it may well be in that Stampede trial, looking at metformin or statins or whatever it might be, there's a particular subset of prostate cancer patients, and actually trying to work out what the biomarkers are that puts people into those categories is what we're interested about. I suppose the one thing that I didn't speak about is there's a lot of people in this field trying to use bioinformatics as to gain the sort of best predictive analytics to sort of, I suppose, guess best at which treatments are going to work for which patients. And our commitment is that, you know, best place science is fantastic. It's a great starting point, but actually getting the sort of empirical data back from patients, both in terms of clinical outcomes, you know, imaging, tumor markers, overall prognosis, survival, etc., but also trying to collect longitudinal molecular data. So looking at how the genome, the transcriptome, the proteome, the immunome change over time is going to be the really vital thing. That's going to be the thing that is critical for us learning off every single patient and understanding which subgroups benefit from which treatments.
How do you manage the complexity of all that? I mean, I saw that list and you had whatever it was, 10 or more different mutations and 10 or more different treatments and caffeine and this and that. So first of all, you have all the omics data. You have to put it together into a comprehensive picture about what's the right thing. And then you have, it looked like, you know, half a dozen or 8 things. So is that now a combination? And if so, how do you dose it? It just seems there's a great deal of complexity because you're exposing so much data. There's a great deal of complexity in all that.
Trav, you want to take that, or do you want to?
Oh no, that's right up your alley, Pat. With pharmacologically, I will say this: we do have a lot of natural products that surface, and again, it's just simply the nature of the molecules. They're small, planar molecules that bind a lot of things. We don't make any claim into the bioavailability, which is always a problem with natural products. However, there are a lot of patients that are taking supplements anyway, and if you can reduce that list to the ones that may be the most effective, we want to do that for their benefit, and then it falls on the clinician. So I'll let Pat take over from there.
Yeah, I think the important thing for me, as someone who's crossing from the research into the clinical world and does both with both hats on, is that actually the clinician's judgment is really important. We have patients that we'll prepare a report for, and And the clinician will say, well, that's all well and good. I understand what you're saying, but I have treated, my lung cancer patients with this combination of drugs. So I'm going to incorporate some of your research, but I'm also still going to give them this drug, or I'm going to do this, or I'm going to tell them to do this lifestyle intervention, whatever it might be. And that's really important for us, actually, because we are trying to get to the truth in each patient and overall. And the science is incomplete. The peer-reviewed science is incomplete. The empirical data is nowhere near complete either. And so actually, the fact that we could give the same report to 3 different clinicians and get 3 completely different intervention strategies out is really important for us building the most solid algorithms. Because otherwise, if we were, it's one of the reasons that we did not set ourselves up as a clinic. Because if we did the research and then almost marked our own homework by treating people in a really specific way, it would take us a long time to improve our algorithms. Whereas actually, by divergence, getting different doctors to use different protocols, different dosing strategies. I appreciate exactly what you're saying, Brad. There's a lot of noise there. I completely get that. But one of the things that we are looking to do, and we are starting at the moment, is a longitudinal registry study, working with a specific group with a large number of clinicians, so we can try and ring fence the amount of divergence, but still have individual doctors making individual decisions.
So just, I'm a process consultant. And so just understanding how this works at a process level, at a workflow level, timing could be really important here. So when I sit down with my clinician, which I will be doing tomorrow, and we have a consultation, things that come into that conversation are valuable. If it takes 5 to 7 days to order your report, and then it gets into the next conversation, but a decision may have already been made about what the treatment protocol is going to be. So how have you found that integration of your advice, which you said takes 5 to 7 days from getting the inputs? How does that fit into the flow of the decision making of the patient working with their medical team?
Yeah, so I think as to where we are now, it's taken us a year of optimizing the bioinformatic pipeline to get where we are. And there is still a manual part of this process, which is, our scientific team sitting down with a long list of everything that's come out, from the large language model, and curating that. And that takes some time. It takes probably on average, still 8 to 10 hours per patient to do that. And as we build the bioinformatics out and we get more quality data and we're able to stratify by, different measurements, we are very confident that we'll get to a process that is almost completely automated. And then at that point, we'll be able to use this as a clinician interface. So, the hope is that the clinician will be able to use this in real time without having to, have the clunky process of getting an NGS report, sending it through to us, getting the report back and dealing with it. Because that's ultimately what we want, Brad. We want you as a patient to turn up. Often no one needs to know about what Astron are doing. What we really want is the data. The clinician can use our software, use our technology, and very much as a peer-to-peer research tool, not as a software as a medical device tool as yet, but in order to make the right decision for the right patient at the right time.
Got it. Thank you. I'll choose one more question from the chat. Rick Davis asks, do you request patients' list of medications to check contraindications, frequently overlooked by HCPs and an important aspect of a personalized report?
Yeah, you're 100% right, Rick, that this is something that we are absolutely going to be doing. The issue with that at the moment is, again, if we start getting into the realms of absolute dosing, we are producing a kind of pharmacopeia encyclopedia, which is a list of all the potential drugs, supplements, nutraceuticals, lifestyle interventions, etc., and sort of potential dosing, etc. But if we get into the realms of directly getting a patient's, blood test results, drugs that they're already on, etc., and working out a strategy, it could be interpreted as software as a medical device right now. Clearly, that is what we are looking to build, but there is a process and a structure to get there in terms of the regulatory side of things. So you're absolutely right, but the way we're getting around that for now is by really handpicking clinicians, working with people that are already doing this, and are used to prescribing some of these drugs already and have experience in doing this. But that responsibility at present falls on the clinician.
Got it. Richard Andrews has raised his hand. Richard?
Yes, hello. That was really interesting. Thank you. First, an aside on the nutraceutical front, which is, as I'm sure you know, it's very hard sometimes to know which particular, nutraceutical actually is meaningful or precisely calibrated. It's really hard to know. You could have 5 versions of a compound you want in a nutraceutical, and one of them actually delivers, and 3 of them have wildly variable doses, and 1 is who knows what. So that might be a service that would be useful for you to help people figure out, but because I don't know if they have good ways of doing that. It's a really interesting problem that you're dealing with in terms of what you predict. I mean, when you're in the world of large prediction of lots and lots of stuff, as you know, you're going to get a lot of noise. And if you're at a place where you don't have a lot of options, even something that doesn't have statistical significance may well be a reasonable choice. But early in the process, a lot of these things, because I think you pulled up something that didn't seem to have statistical significance, at least in one part of the study, do you have a way of helping patients or clinicians figure out, or what is the right way to figure out when you have a vast number of potential interesting results, which of them should be listened to for patients? It probably varies. Early stage patients with lots of good treatments maybe should stick very much with standard of care, and late stage with very few should. How are they supposed to think about it? How would you recommend they think about it? And how do you titrate your results over time to make them better, so that some of the results sort of rise to the top as definitely consider these, and these are not as good, but maybe?
Travis, I'm going to let you come back to the kind of hierarchy of evidence maybe in a second. But the first thing to answer is very smart from you, Richard. You're right. There's an awful lot of noise. And that's why the first thing that we are trying to do is to control as much as we can, right? And so you're right. If we've got patients coming to us with every type of cancer at every stage of their disease, with a variety of different sequencing tests, some with just genome, some with exome, transcriptome, immunome, etc., it's going to be overwhelming, right? So in terms of the first study that we're actually doing, we're actually working with the largest surgical cancer unit in Europe, and we're looking at a very, very sort of bespoke clinical problem, I suppose, which is we're looking at MRD in colorectal cancer. So this is people with stage 1 to 3 colon cancer who have had a curative surgery and possibly some chemotherapy, but they have no macroscopic evidence of disease. Now, I'm sure all of you have heard of, kind of Signatera or circulating tumor DNA testing to look at, people in that curative setting who've still got circulating tumor DNA in their blood. And the risk of recurrence is huge in patients who do have circulating tumor DNA. And the problem with those patients is at the moment, we're spending billions on doing that testing, especially in the US, but there's nowhere ready to go with it. As a surgeon, you're seeing that patient 3 months after you've done the operation. You have to give them this really hard information that they've got circulating tumor DNA and their risk of recurrence is high. You can't operate again because there's no actual lesion. This is molecular disease. You can't irradiate for the same reason. Chemotherapy has been shown to be non-inferior, but no real benefit at this point from the data that's out there. And so there is a big blank space here. And so we are, from the discussions we're having, we're almost a few years too early because people haven't kind of seen the problem with their own eyes yet. But for us, the study that we're doing is trying to drug MRD, which is a clinical setting where we can make the data just a lot more homogeneous. Okay, we've got people coming in. Yes, they've got different genes, different transcriptomes. They've got the same tumor. They're at the same stage of their disease. And we can see in that blank space whether our approach of kind of precision oncology in a safe, tolerable, proportionate manner, drugging genes, pathways, proteins, actually is extending the time to recurrence or even reversing MRD altogether. So that's
So what you've got then, so the way you're doing this trial is not as multiple different endpoints, but as one endpoint with a black box in the middle. The black box is you take the genomic data, and out of the black box, and I don't mean this in a bad way, out of the black box comes a treatment, and that is what the clinical trial is measuring, whether that treatment lowers some endpoint at the end. The endpoint is survival or time to recurrence or something like that, with a very heterogeneous black box in the middle.
Yeah, so I suppose we're treating, I wouldn't call it a black box, but a molecular tumor board. So the interventions can be a huge point of difference. Yeah, the intervention is the molecular tumor board itself, right, which incorporates our software and technology plus clinicians making, well, a tumor board of scientists, clinicians making a decision for that patient, and then the patient going on that treatment. Yeah, you're absolutely right.
But there are, I mean, if you have all those different variables in the middle, it starts to become very hard to control what exactly is going on. I mean, maybe it's the wisdom of the doctor rather than the wisdom of your computer
Right,
program.
but the approaches will be defined. So if someone's got a PIK3CA mutation, these will be the kind of, it's not going to just be Dr. X plumping for something because they read something in, a random journal last week. There will be a process to that, but there will be, as with everything, until we're approved as a software, as a medical device, we can't just say our software is the intervention and that's it. That just wouldn't.
But of course, if you start to stratify, if like in the middle of your trial, you say, well, we work well for the PIK3CA people and we don't work so well for the P10s. Now you're really corrupting the trial. It becomes very difficult to get results because you're going to find subsets galore, some of which work and some of which don't. You have to sort of maintain the integrity of the trial. It's either universal or it's, subtype by subtype. You can't sort of switch between them as you want to.
I think it depends what your endpoint is, I suppose. And I think in this context, this is not, I mean, this is almost a proof of concept study. And then going forward, you may well be right that we will stratify and say, actually, in the people with, TP53, APC deficient colorectal cancers, we're going to try this intervention, which is born out of this research and our engine, and we're going to see if that can extend the time. So you may absolutely be right. But I think at this point, the approach is so novel. And the other thing to say, I suppose, is that measuring the data longitudinally, so actually not just focusing on the MRD status and the circulating tumor DNA status, but also looking at the mutational burden, the transcriptional burden, what's changing in terms of the expression in, someone's got a PIK3CA mutation, what's happening to PI3K and AKT at a transcriptional level, for example. That is also incredibly important. And that's a part of this that I think is being missed, where we concentrate solely on endpoints, clinical endpoints, does the patient, what's the disease-free recurrence level and what's the overall survival? That's obviously the most important thing. But also looking at, what's the intervention? Did this patient have the effect on PIK3CA that patient did, even though they both took the same dose? Or did we not drug it hard enough? And was there a reason for that?
It's ambitious. It's really neat. I wish you the best of luck. It sounds very interesting.
We hope so.
Yeah, your first question, Richard, really is the heart of what I think about all the time. And I think the best way to answer that is the rigidity of the system, as we all know. There's very defined criteria for which drugs enter the toolkit of an oncologist. However, patients have much different calculus, as you mentioned, that especially with nutraceuticals. You go down that evidence chain and then the lens becomes, what have I got to lose when something is extraordinarily safe? And so we want to provide that, even though it's down the evidence chain, and they can make that decision with their clinician. But the high tier evidence, like the celecoxib study, they absolutely, that will be ranked as a higher, that they need to pay attention to this one.
I had a related question, which is confidence. So you can have recommendations that you are very confident about and other recommendations which are less confident about. Is there something in your recommendations where you share the confidence factor you have on that recommendation?
Right. We iterate this product all the time. And right now, we have it's linked to references, so the clinician can easily see the data, and it's flagged if it's clinical trial evidence. So it would be difficult for us to give a confidence because we don't know. You see a lot of that with trials. You would expect some of these trials, and there's been hundreds of these trials that look at things like metformin or beta blockers adjunctively in oncology. When you expect to see results, sometimes you don't. And we don't know why. The complexity of the human body, the interaction with the immune system. So that sort of evidence is obviously top tier because we don't know. However, when you go down with the risk reward profile, and when there's 200 repurposable drugs, we want to give the ones that present the best chance.
And I think the
lower
other,
confidence ratio. Yeah, sorry, go ahead, Ben.
sorry, Trav. I think the other thing is that we are really prioritizing the empirical data that we get back, Brad, as well, right? So it's all well and good for us to have scientific confidence based on a clinical trial. A lot of those clinical trials, as you know, may not have been substratified in the way that we would like them to have been, or that we might find as powerful in the future. But the empirical data and the way that we're collecting it, we hope is going to be a game changer. I think the other thing to say is that our use case scenario for molecular testing is unique, right? So normally you go to your oncologist and you have a molecular test, even if you had infinite money to spend, or your insurance company was very generous or whatever, and you could get a molecular test every 3 months. And often the oncologist is looking for 3 or 4 genes in a specific cancer type, which might mean that they can make a recommendation or not. And the evolution of that is not going to be every 3 months in most cases. Whereas for us, because we're able to action so many more changes at a genomic, transcriptomic, proteomic level, actually there is a real use case scenario for testing much more frequently. And that testing is incredibly valuable, because what we are missing in cancer now, cancer evolution, is that we've got, even if we look at CBioPortal and TCGA and all that, and we look at the evolution of something well studied like colorectal or lung adenocarcinoma, and we look at what happens during the course of this disease, and you're diagnosed and you've got KRAS and you pick up TP53 and then APC or whatever else it might be, that data comes from huge numbers of patients who happen to have had molecular testing at a different point in their disease course. So there is, as you were talking about earlier, Richard, there is a huge amount of heterogeneity and noise in that data. And what we're trying to do is to really find a good use case scenario so we can map longitudinal patient data in thousands of patients in each cancer subtype. So within colorectal cancer, there are 7 or 8 identified subtypes, and being able to map what happens at various omic levels so that we can get the sort of predictive analytic capacity to be able to say, well, we think this is going to happen next because that's the trajectory that we've mapped out, and we're going to try and drug that one step earlier. That's where we think there is real value for doing this.
Russ asked a question about your testing inputs, and he says, are you using whole genome sequencing, whole exome sequencing targeted, like a Garden 360, or all the above? Do you ever go back to the patients and the doctors and say, there's a hole here, you really should get this kind of test to fill out the picture?
It's a great question. And at the moment, as I said, we're not a testing company at present. And so we work with whatever the patient has. There's a few things to consider. Obviously, temporal. I had a patient very recently who had a Guardant report done in January, which showed a PIK3CA mutation, a CDH1 and ESR, and had done nothing apart from go on a ketogenic diet for 6 weeks. And not only had the mutations disappeared in both a Guardant and a Datar report, but when we looked at the transcriptome, PI3K and AKT was also downregulated, which was super interesting. And she'd done nothing else apart from a really aggressive ketogenic diet, no other treatment whatsoever. And that is the power of the longitudinal analysis. You're absolutely right, though. So the temporal consideration is one thing. Is the report that you're giving me 2 years old? Have you had 3 rounds of chemotherapy in the meantime, or some immunotherapy? We often find that doctors often think that more is better. So the number of doctors that order whole genome or whole exome just because they think it's better. And I actually, sometimes think, well, the read depth might not be so great for this. So we might miss some of the lower variant allele frequencies in terms of mutations at a sort of 3 to 5% level when we do a whole exome or a whole genome rather than a sort of just a pan-cancer screen. So there isn't a definite answer for that, Brad. From my point of view, I like as much breadth as possible. So I like to know what is going on at a genomic level, at a transcriptomic level, at a proteomic level, if possible. I know you guys have done a lot of talks about this in the past and are well versed in that as well. So I'm not going to bang on about that, but we see a lot of patients where there is not a druggable KRAS mutation or an EGFR, and then we go and look at the transcriptome, and it's really clear that there's a massive overexpression of KRAS in the transcriptome that we would otherwise have missed if we'd just done a genomic analysis. So yes, we do advise patients about that, but we're also really aware, obviously I'm based in the UK, so I'm really aware that everything is out of pocket. So there is that really fine balance of trying to get the best information, the most contemporaneous information for the patient at the right time, so we can make the best decisions or the best research, but also understanding that cancer's an expensive game. So.
Okay. Russ also asks, and this may be similar to questions we've already asked, are your suggested therapies validated options or emerging mechanistic? I haven't found enough of the validated options. We've been forced to look into non-validated predictions.
I suppose that comes down to what you call validated. If you call validated something FDA approved, then we do look into things that are FDA approved, of course. But generally, if someone's had next generation sequencing, if you've had a Tempus or a Boston gene or a Guardant report, and you've got a mutation or a fusion or something that there's an FDA approved therapy for, that'll be on your report anyway. So that's fine. A lot of what we look at is either oncology drugs off label, so if you've got a mutation, you've got a HER2 and you've got a cancer where there's no study on looking at Herceptin or another HER2 blocker, and we think that might be a good strategy in your particular case. That's non-FDA approved, but from a scientific point of view, there is validation at blocking HER2 with Herceptin or an associated drug in a different tumor type. So we look at both to answer your question, Russ.
Okay. Alan Morris has a question really about, I would almost say, competition. He says, I have the following somatic mutations in my bone: metastatic prostate cancer, CDKN2A, CDKN2B, MTAP, PTEN, FAST, MAP3K1. How is your approach better than me asking ChatGPT or other more clinical medicine-centric AI engine for the most promising phase 2 or other, for example, observational studies such as the coffee study you presented regarding ER plus tamoxifen treated breast cancer?
Trav, you want to take those, or you want to take this?
You can, absolutely. And in the base level, that's what we're doing. But we have a team, we have a methodology and approach. We hope we don't miss anything. And there's a lot that goes into it. I mean, ChatGPT is good, but you have to be very careful. But like Pat said in the opening, any clinician could do this with a lot of time per patient. But once we get, and the beautiful thing about the database is it just gets better and better and more refined over time.
Yeah, I think the way that we do this is important. You're right. The technology is there for you, Brad, to go and do this. Of course it is. But it's taken a long time because at the core of the approach is obviously a bespoke LLM, which is currently trained on over 100,000 peer-reviewed papers through a variety of open source data sets. And we have a weighted evidence system that uses loads of inputs: disease specificity, study design quality, preclinical binding affinity, clinical validation. It incorporates, all the omics that we can. We use a process called named entity recognition to extract really meaningful associations at a sort of drug pathway, gene stage. And it's a sort of very multi-dimensional scoring system. So there's a sort of literature quality score, a sort of mechanistic plausibility score, a disease context score, and then a kind of safety overlay. So if there's anything negative, we'll flag that as well. And we'll say, well, you've got a PIK3CA mutation. We might recommend celecoxib or, a ketogenic diet, but actually you've also got this. and we think that, fat metabolism might be really important, and therefore a ketogenic diet might not be the best, answer for you to cover your whole disease. So I think that's what makes this different. And I've had a lot of patients that have come to me who have put in their whole exome report or NGS report into GPT and asked GPT to come up with a repurposed drug protocol. And honestly, it's hard because you can make anything make sense on a piece of paper, right? If it's compelling enough. But some of the things that have come out have been actually quite scary and could have done a lot of harm. And I mean, not just a lot of harm from a sort of elevated liver function tests or, some side effects, but actually do a lot of harm in terms of the disease itself.
Russ is coming back to the testing question, assuming price is no object. Would you recommend WGS, WES, proteomics, RNA? You mentioned quite a number of omics. Are there any ones that, if price were no object, and I guess what you said is with breadth, so you would be looking to fill in holes that they have WGS, but they don't have RNA, et cetera. So how do you think about what are the principles you're applying to what tests people should get?
Yeah, it's a really good question because we obviously know that non-coding regions that you get in the whole genome are super important as well, but you get that information. So the way I think about it is the more breadth that you get in terms of genomic sequencing, the less read depth that you get. So you might get more of the non-coding regions, but you might miss a mutation that is actually driving the next iteration of the cancer. You might just miss that from a sensitivity point of view. So I think from where we are now, I generally go for a pan-cancer panel, which is somewhere generally between a sort of 500 and 650 gene panel. It's, and this is irrespective of cost, alongside with full RNA sequencing, and if possible, proteomics, but if not, at least some immunohistochemistry, together with all the usual kind of PDL1s and MMRs and CLDN18s and things like that. I think when we get into the remit of doing specific that's our professor calling. If we get into the remit of specific immunogenomic assays, etc., I think, if I'm honest with you, with where we are right now, we haven't quite got the data to be able to work with such huge amounts of input. So I would say pan cancer, 500 to 650 genes, full transcriptome, plus some immunohistochemistry or proteomics is where I would sit. And I think, there are tests that do that in full. I know that you guys are big advocates of the tumor portrait, Tempus do variants, etc. There's a lot of very good companies doing lots of good things.
Let me just ask a quick question about keeping your data current. So we had Mark Taylor and Gabriel Gavazzi on, and they are doing something similar, and they're saying they're checking out 1,000 research papers a week and adding that into their, the inputs into the decision making. How does that look for you in terms of keeping up with all of the new studies that are being reported all the time?
Yeah, that's the biggest part of our spend at the moment is the GPU spend on doing exactly that, and then manually also going and making sure that is happening robustly and that the model is iterating and improving over time. But as a clinician, frankly, again, I go back to the quality of how we pre-trained the LLM in the first place, and having the right scientists. The people that we've got working on this are ex, Sanger and Cosmic scientists who understand this, and that is our core. We're not a team of people that have never worked with cancer before. We understand the heterogeneity, the complexity, and we really respect the disease and the complexity, and the fact that we are nowhere in terms of being able to be sure about what we're doing right now. I think that will stand us in good stead.
Rick Davis asked something we should have done earlier. I'm sorry we didn't, and that is about access and cost. So if people are persuaded by Astron Health and what you're offering, they want to go to their physician and say, can we get this input into my care strategy? How would they go about doing it? What would it cost?
Yeah, of course. So in the US, it's slightly different for regulatory reasons. We sell exclusively through clinicians. Now, that can be your primary oncologist. It can be another clinician that you're using with an expertise in genomics. There are lots of metabolic or integrative physicians or even NDs around that we can work with as well. If you don't have a physician that is prepared to refer you through, then we can obviously, work with you and refer you to one of our partners. Once that process has happened, we will then set up a free call with myself to take you through the process, explain exactly what you're doing, have a look at your reports, explain what the research would look like, what we're hoping to achieve, how long it will take. The cost typically to the patient is in pounds, it's £950 at the moment. As we scale and the report generation becomes more automated, two things will happen. Obviously, the cost to the patient will go down significantly, but also we're hoping to semi-automate this so that this can be a clinician level function, so that the cost will be attributable to the clinician and the patient will be able to just receive the report as, one of the bonuses of working with that clinician. So that's what we're hoping to get. But at the moment, the cost in the US, I think is, correct me if I'm wrong, Raf, I think it's $1,250 direct to consumer at the moment. Yeah.
It's closer to 1,000 or 150 actually.
Sorry, it's gone down 1,100. And if you want to sign up, there's please just visit astron.health, book in a call with myself, Travis, one of the team, and we'd be absolutely delighted to take you through the process individually. We're a small team, but one of the benefits of having a software business is that we can stay lean and be really personal. So we're very happy to do that.
How would you define the persona of the providers you're selling to? I can imagine that if there's a oncologist at an academic research medical center, they probably think that they know it all and there's nothing that you can add. If I were a community oncologist, perhaps I don't, and I'm treating multiple kinds of cancer, I might have some humility and think that there might be new stuff that I don't know about. So it'd be useful for me to get your service to kind of as an aid to that decision process and making sure that things are current and I'm not missing something. So how would you describe the different personas you're selling to and providers, and what are either the objections or the receptivity you're getting?
Yeah, I think I would have said the same before we started this, Brad. But the first thing to say is that a lot of conventional academic or non-academic oncologists in the conventional system have their toolkit. And even if you provide them with compelling data for a repurposed drug, some of them will be reticent to prescribe that because it's outside their wheelhouse, and I completely get that. However, within the context of a clinical study setting, what I would actually say is that the more academic doctors, they see this as a massive opportunity. When we stop talking about, it's really the way that you frame it, okay? When we stop talking about drug repurposing and we start talking about synthetic lethality and drugging MRD, everyone stands up and they get very, 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 very excited about this because it's something that they could do now, which they haven't even thought about. And the other way that, so I think actually the people that are most engaged from our point of view, we've got a variety of academic professors, assistant professors, etc., who are really open-minded. And a lot of them have had research funding withdrawn. It's much more difficult to get research funding post-COVID, and obviously with what's happened governmentally as well, it's been tough across both sides of the pond. In terms of the people doctors that we're selling to directly now, being honest, the majority of them are people that are integrative cancer specialists. So a lot of them are oncologists who, in addition to practicing conventional oncology, have set up clinics alongside that where they can address these alternative strategies for their patients in addition to standard of care. So they are cancer specialists, but they have expertise and awareness of using adjunctive drugs, supplements, dietary strategies, etc.
I just noticed that Will LaValle joined. I just posted just a couple of hours ago the meeting summary from the session we had with him, where he was talking about molecular integrative oncology, many of the same molecular kinds of pathways you were talking about and the algorithms you're talking about. So he was working in the same domain. If you don't know him, you should, and I can connect you outside of this.
We know Will well. It's a small
I've been
world.
following Will's Will's work for a long time.
Okay.
Hi, Will.
Hi, Will. He's a great guy.
Well, I'm glad to be here. Very interested in absolutely the field is emerging, and there is a newer, I think, openness to the understanding that these repurposed drugs are multi-purpose drugs, and that we have data that can support the use in addition to conventional oncology treatment. And so, absolutely, and it's good to be here.
Thank you, Will. We're past the hour. I should start to wrap us up. I always like to leave an opening at the end for the discussion leaders to leave us with kind of key parting words of wisdom or conclusions or main messages. Would you like to each take a moment to do that?
Yeah, sure. Trav, you want to go or shall I go?
Oh, go ahead. Go first, Ben.
Okay, no problem. So I think the main thing that I want to say is that we want to make precision oncology equitable. We want to make it scalable, and we want to make it as impactful as we can today across the globe. We don't want to wait for future drugs or future technologies. Those will come and those will be absolutely vital. The patients I'm seeing are getting younger and younger. We're not talking about, or we shouldn't be talking about 5-year survivals anymore. We need to be talking about 20-year survivals. And I think the opportunity and the data that we have at our fingertips gives us a massive opportunity, a massive public health opportunity to scale precision oncology right now. And I think just the two things that our two core values are patient centricity and collaboration. So we really want to build an infrastructure that groups like yours, people like Will, theory makers, the Mark Lintons of this world, etc., can use the data that we have to go further. We don't claim to have all the answers, but we want to try our best to enable smarter people than us to get there.
Yeah, thanks, Brad. And I think you're a process engineer. I think what when I look at the healthcare system within this realm of oncology, what you notice is a vast underutilization of the pharmacopeia. And it comes to pharmacology where on average, these small molecules affect a dozen relevant cellular pathways, but due to financial reasons, they will never get FDA approval. There's a perfect case of that recently with statins, where they've noticed a linkage between APC mutations in colon cancer with a protein called RAC1, which downregulates this incredibly important oncology pathway, WNT. They are developing de novo statins to do this, which will cost, again, billions, and then be, typical drug prices where we have statins existing today. So it's a problem. It's a systemic sort of failure in the system, the way the system is structured. And the same thing with data. The same thing with these genomic reports are vastly underutilized. And so from a patient-centric view, we're trying to fill that void.